Good afternoon, I'm Sherry from Shenzhen.
A third-year PhD student in Dr. Ray Chen's lab. It's my honor to introduce our next speaker, Dr. Hongkui Zeng, Executive Vice President and Director of the Allen Institute for Brain Science. Dr. Zeng is internationally recognized for her leadership in developing high-throughput, large-scale, open-access datasets and tools to accelerate neuroscience discovery. Since joining the Allen Institute in 2006, her work has led to widely used resources such as the Allen Mouse Brain Connectivity Atlas, the Common Coordinate Framework, and the Brain-Wide Transcriptomic Taxonomy and Atlas. These contributions are now foundational for characterizing brain cell types and understanding their functions in health and disease. Dr. Zeng received her PhD from Brandeis University, where she studied the molecular mechanism of the circadian clock in fruit flies. As a postdoctoral fellow at MIT, she studied the molecular and synapse mechanism underlying hippocampus-dependent plasticity and learning. Her contributions have been recognized with numerous honors, including the 2016 AWIS Award for Scientific Advancement, the 2018 Geotransformative Investigator Award, and the 2023 Prado Research Award, and an elective member of the National Academy of Science and National Academy of Medicine. Please join me in welcoming Dr. Hongkui Zeng.
Okay, thank you very much, Sherry, for that nice introduction. I also want to thank the organizers for including me in this symposium, speaking among this, deeply honored about speaking among all these amazing speakers, the true trailblazers of biomedical research. I also am very happy to have the opportunity here today to share some of our latest work with you. The collective goal of neuroscience is to understand how brain works at all these different levels, from cell circuits to computation to the generation of behavior and cognition. This is a daunting task given the extreme complexity of the brain with millions to billions of cells, neuronal and non-neuronal, across a variety of species, their central nervous system. And also thousands-fold more connections among the neurons, as beautifully shown by Tom in the previous talk. To tackle this complexity, we believe it is essential to take a multi-level approach, studying cell types, circuits, in vivo physiology and behavior, and perhaps eventually to achieve a computational understanding of how brain processes information and generate behavior. And also through the process to extract organizing principles of the brain at all these different levels. At the Allen Institute for Brain Science, our primary focus is actually just on the first two levels of the brain, trying to understand the diversity of cell types and also how they form functionally specific circuits, with a primary focus in the mammalian brain from mouse to human. We take a big science, open science approach, generating large-scale comprehensive data sets describing the properties of cells and circuits, striving for completeness, and present the data as foundational resources to the community. At the same time, we also try to use a data-driven approach to extract rules and principles underlying those data and observations. So with that kind of motivation, we have established multiple high-throughput technology platforms at the Institute. Our front-end approach is the highly scalable single-cell genomics approach, including single-cell RNA sequencing, single-nucleus ATAC-seq, and spatially resolved transcriptomics. Through the use of those approaches, we profile hundreds of thousands to millions of cells and use them to classify the cells into different types, understand their relationships as an initial approach in understanding the diversity, the complexity of the brain. We also continue to make genetic tools targeting those specific cell types Traditionally, we have been generating transgenic mouse lines using specific marker genes and things like that. In recent years, we have been trying to take advantage of the combination of single-cell RNA-seq and single-nucleus attack-seq data to specifically identify putative enhancer elements that are specific to certain cell types. And then we use a viral approach such as AAV to test the specificity of cells. And we have been able to achieve quite a good success in identifying specific enhancer AAVs. At the anatomical level, we use large-scale light and electron microscopy to obtain and describe, reconstruct the full morphologies of neurons and their brain-wide projection patterns, as well as synaptic level connections, and use those information to understand the properties of cell types as well. To bridge molecular information and anatomical information, we use a set of multi-model approaches, such as triple modality patch-seq, as well as bar-coded connectomics, to relate molecular identities obtained from our transcriptomic studies to the structural and functional properties of the cells. And finally, in recent years, we have started to build technology platforms for proteomics as well, profiling both the quantity and the nature of the proteins, as well as their spatial distribution patterns, as a bridge between genetic and gene and RNA information with the structure and function of the brain. Finally, underlying all these different technology, experimental technology platforms, we also build strong computational approaches for large-scale data management, analysis, especially integrative analysis, and finally, dissemination of all the data and knowledge and tools to the community. Today in my talk, I will just focus on telling about our series of studies and a few recent examples in using the single-cell genomics approaches to arrive at an initial version of the atlases, taxonomy and atlas of all the cell types in the brain. And also, in addition, importantly, learn what are the underlying rules govern the diversity. And then begin also to use the foundational knowledge of the cell types to begin to understand how the cell types change under different conditions in development, in aging, and under a variety of disease conditions. And we're just, this is really just the beginning of the journey. Okay, so our starting point in understanding cell types actually is from a single brain region, in particular, a single cortical region. And I just want to use this study as an example to illustrate what we have learned. This is a work in collaboration with many groups in the Brain Initiative Cell Census Network, BICCM. In this particular study, we used the multimodal approaches mentioned in my previous slide to study cell types in the primary motor cortex across 4 mammalian species: mouse, macaque monkey, marmoset, and human. And by integrating the data all together across modalities and across species, we arrive at this consensus cell type taxonomy with about 100 cell type, transcriptomic cell types described for the single cortical region, glutamatergic, GABAergic, and a variety of non-neuronal types. In addition to creating this atlas, we also learned several important principles. We find that the cell types are organized in a hierarchical manner. We can classify them at different levels of granularity, from class to subclass to type. And we are able to achieve multimodal integration across the variety of cellular properties and modalities. We observe the coexistence of discrete and continuous variations of the cellular property, painting a highly complex cellular landscape. We also observe coexistence of cross-species conservation and divergence. With the conservation mainly at the class and subclass level, all the major cell types are conserved, whereas the divergence appears at a specific type or cluster level, and also at gene expression, individual gene expression level, with about, for example, 20% of genes differentially expressed between different species, even in conserved cell types. Here is another example of a multi-model integrative study in which we use a patch-seq approach to profile a large number of GABAergic interneurons in the mouse visual cortex. And by integrating morphological, physiological, and transcriptomic information together, we define what we call the MET types, M-E-T types. And the MET types and all the GABAergic MET types are shown here. There are 28 of them, grouped into the well-known MGE-derived somatostatin and parvalbumin types, and also CGE-derived VIP, multipolar, and neurogliaform types. We find that the MET types have intermediate granularity compared to T types, but they have higher cross-modality correspondence and mutual predictability. They also recapitulate traditionally defined interneuron types very well, as shown by the names assigned to those different MET types, especially the somatostatin types, and provide additional subdivisions primarily driven by layer specificity of the different GABAergic type, which is an interesting observation that transcriptomic profiles really specify the different layers where the cell types are located. So in the end, we find that the MET types can serve as Rosetta Stones, again, to bridge molecular identities with cells profiled in other kinds of data that actually lack molecular information. For example, assigning molecular identities to cells reconstructed from electron microscopy data through morphology matching. Okay, so fast forward 2 years. The field is moving really fast, again, in collaboration with the BICCN consortium members. We have then scaled up the single-cell transcriptomics and spatial transcriptomics approaches to profile millions of single cells in the entire mouse brain. And by integrating those two different data types together, we generated this high-resolution transcriptomic and spatial atlas across the entire mouse brain with more than 5,300 clusters identified. And we grouped the clusters into a hierarchical structure, supertype, subclass, and class levels. And from this effort, one striking finding immediately jumped at us is the extraordinary diversity of the neuronal types. Neurons form 5,200 out of the 5,300 clusters that we obtained by an unbiased clustering approach. And we also find that the neuronal types are driven very much not only by the molecular identities of the cells, for example, the neurotransmitter type, but also very much driven by the region where they come from. And then furthermore, after setting up this cell type taxonomy and atlas, we obtained a whole set of gene co-expression modules, cell type specific gene co-expression modules. And we found a particularly large set of transcription factor gene co-expression modules, as shown here, 52 co-expression modules that have this amazingly specific cell class, subclass, or type specific expression pattern. And collectively, we believe that forms a transcription factor code that defines cell types across the entire adult mouse brain. All the neuronal classes as well as all the non-neuronal classes. If we dive a little deeper, we divided up the 5,300 clusters into 7 neighborhoods. They are not mutually exclusive, so we don't put them as a level of hierarchy. It's just for the operational sake of analysis. 6 out of the 7 neighborhoods are neuronal neighborhoods, and they are organized by major brain structures. So we can look a little bit deeper This is just one little step deeper. Of course, we have gone a lot more deeper later on, but I just wanted to show you here. Here it shows the first three major neuronal neighborhoods, glutamatergic cell types in the entire pallium or talencephalon, the cortical structure of the talencephalon, shown here. And here is all the GABAergic neuronal types within also the entire talencephalon that are generated in the developmental subpallium structures. And here, this UMAP shows all the glutamatergic and GABAergic neuron types within the hypothalamus, and its closely related amygdala areas, extended amygdala areas. In each case, we see not only this extraordinary diversity of transcriptomic profiles and their intricate relationships, but also the exquisite correspondence between transcriptomic specificity and spatial specificity as shown in this Murphy spatial transcriptomics data. So similarly, here are the other three neuronal neighborhoods. This one is actually the simplest. This is a neighborhood for thalamus, excitatory projection neurons in thalamus, and epithalamus, which is the lateral and medial habenula. And then two highly complex neighborhoods for the glutamatergic or GABAergic neuron types in midbrain, hindbrain, and cerebellum. Again, in each case, we see this beautiful correspondence between transcriptomic specificity and spatial specificity. Another interesting finding that from this kind of analysis is a dichotomy of the brain that we observed, meaning that in the ventral part of the brain, including midbrain, hindbrain, and hypothalamus, we find that the transcriptomic clusters are all, there are numerous transcriptomic clusters, and each of them is also quite small. You know, they contain a small number of cells. And spatially, those tiny but numerous transcriptomic clusters are spatially localized to specific nuclear structures and subnuclear structures. The dorsal part of the brain, on the other hand, including the entire cortical structures, and also thalamus and cerebellum, there are much fewer number of clusters, but each cluster contains many more number of cells. And those clusters tend to have much wider spatial distribution with further gradient distribution of gene expression within each cluster. We think this kind of regional brain, regional diversity, the dorsal part versus the ventral part of the brain, may be reflecting a differential evolutionary origin, with the ventral part of the brain being much more ancient, being like the first part of the brain emerging in the vertebrate, and that are important for the survival function of the animals, such as metabolism, feeding, reproduction, and instinctive behavior. Whereas the dorsal part of the brain is evolutionarily new, and they are primarily responsible for adaptive behaviors such as sensory motor specialization and cognition that allow the animals to adapt to their local environment better. And because they are evolutionarily sooner, their cell types are not segregated that much anymore. However, it's the structure that undergoes rapid evolutionary changes. So we see all this widespread expansion of the regions and also the emergence of many different axes of gene expression gradients. So again, this is just an example to show that what we can learn from glean at even the history of the cells just from the adult stage comprehensive transcriptomic profiles. So diving a little bit further, I want to use one particular type class of neurons as an example to illustrate the diversity and other interesting properties. And this is the dopamine D1 and D2 medium spiny neurons in the striatum. The D1 and D2 medium spiny neurons form two major subclasses, as shown here, D1 and D2. So they are well known, they are intermingled in the striatum. However, transcriptomically, they are highly distinguishable. We also find many clusters within the D1 and D2 populations. And interesting from this constellation diagram, which is extraction of the UMAP by plotting the centroid of every cluster as nodes and the edges reflecting the relationship, similarity relationship between the clusters. We find that the clusters of D1 and D2 are also intertwined, suggesting a close transcriptomic relatedness to each other. So where are those clusters? And if you look at the spatial transcriptome merge data, you see these are all the D1 clusters, and these are all the D2 clusters. They form this beautiful parallel spatial organization, occupying different domains of both the dorsal striatum, medial lateral, dorsal ventral, things like that, as well as the ventral striatum, which is the nucleus accumbens, and olfactory tubercle areas, with highly specific clusters, specific to, for example, the core and shell of accumbens, anterior, posterior, medial, lateral, all these different domains. So the transcriptomic clustering is able to tease apart the spatial heterogeneity of the cells within the D1 and D2 MSM population. And then furthermore, I also identified two specific gene modules, the light blue one and the dark blue one, that coexist, that exist in both D1 and D2 cell populations in the same kind of a gradient distribution. So collectively, those two gene markers define the dorsolateral to ventral medial axes, gradient axes across striatum in opposite directions for both D1 and D2, suggesting that it's this kind of a gradient spatial transcriptomic map that defines the heterogeneity, spatial heterogeneity of D1 and D2 cells at this very fine-grained cluster level. Okay. So I talked about the extraordinary diversity of the neuronal types. There's not so much diversity in the non-neuronal populations. There are only about 100 clusters out of the 53,000. However, we still observe interesting diversity in the non-neuronal population, particularly in astrocytes and ependymal cells. Both are neuroglia cell populations generated from the same progenitor cells that generate neurons. So here are the UMAPs showing all the astrocyte clusters and all the ependymal cell clusters. And if you look at the spatial distribution, again, the transcriptomic clusters corresponding beautifully with the spatial specificity. There are three major types of astrocytes. The dominant type is the gray matter astrocytes that are intermingled with neurons in telencephalon and all the non-telencephalic structures, and they have broad regional specificity. The second major group of astrocytes are the interlaminar astrocytes, or they have other names as well, that actually are found along the pier surface, both external and internal of the brain, and that form this kind of a barrier between brain and blood and peripheral system. And the third major group of astrocytes are those specifically lining the rostral migratory stream, which forms a tunnel that allows the adult neurogenesis generated olfactory bulb granule cell precursors to migrate through the stream from the dorsal subventricular region all the way into the olfactory bulb. The astrocyte clusters, the individual clusters forming this trajectory aligns perfectly with the spatial anatomical trajectory from subventricular zone all the way into olfactory bulb. Similarly, for the ependymal cell populations, we might find many, many clusters, subclasses and clusters. And those clusters are specifically located in different parts of the ventricles. Anterior, posterior, medial, lateral, dorsal, ventral, and as well as all the circumventricular organs. And each cluster is found in a specific location of the ventricular structures. And here I just wanted to specifically highlight a special kind of ependymal cells called tanycytes, because I will talk about them later. The tanycytes are lining the ventral third ventricle in the middle of the hypothalamus, and also this circumventricular organ called median eminence. And the transcriptomic clusters align very well with the traditionally defined tennisites, alpha-1, alpha-2, beta-1, and beta-2 tennisites based on structural and morphological and molecular analysis. Okay, overall, putting all this together, we, in addition to publication of this atlas, we have now built an online resource called Allen Brain Cell Atlas, or ABC Atlas, to display the whole brain scale single-cell RNA-seq data and spatial transcriptomics data in an online interactive manner to allow users to explore all the different cell types at different levels, as well as the expression patterns of all the genes in the mouse genome. Furthermore, we have developed a computational tool called Map My Cells. And that is a cloud-based tool to allow users to upload their own single-cell RNA-seq data, map every single cell in their own data into our whole mouse brain reference cell type atlas to obtain a cell type identity from our reference, and so that you can compare with your own clustering result, and also you can obtain a spatial location of that cluster. And we are in the process of continuing to improve this computational tool. Okay. So with that, after creating this whole mouse brain cell type atlas across the entire brain, we moved on to answer the next level question is, where does this extraordinary diversity of cell types come from? How are they generated? How they emerge during the developmental process? So then we go back now, going to a single cortical region, mouse visual cortex again, and we developed a high resolution, this time high temporal resolution transcriptomic cell type atlas for this one single cortical area. So basically we sampled 35 time points across development from embryonic E11.5 all the way to P28, and then combined with our adult data with profiling every single day, sometimes even twice a day, from E11.5 to P17, and then every couple of days after that. And of course, as you know very well, many developmental events happening during this period, from neurogenesis, gliogenesis, to synapse formation, as Tom talked about, to experience-dependent plasticity, refinement, remodeling of the circuit, all happening during this period. So what do we find from the transcriptomic atlas? So here is a UMAP derived from this data set, which is just a little over 500,000 cells profiled, high-quality cells passing stringent QC. And this UMAP is colored by the different cell type identification, class, subclass, clusters, as well as by the ages of the cells profiled. And I wanted interesting thing, observation from this, just an initial view of the cell type atlas is this particular time point. I want to tell you, highlight the perinatal time point from E18 to P0, where they are in the UMAP. And by color, they are right here. So this is the transition point from largely neurogenesis, generation of all the major neuron types, to post-mitotic differentiation of those neurons. And from the UMAP, you can already see that there's a striking diversification of transcriptomic profiles after the cells have exited mitosis. So there's this diversification of transcriptomics among the individual neurons as they gradually mature and as the circuit forms. Okay, so here, first I'll talk about the prenatal part. We generated this special UMAP that's basically covering mainly the prenatal time points with a little bit postnatal. And from this, we constructed this subclass level developmental trajectory tree that covers all the cell types in the visual cortex. And just wanted to quickly point out, orient you a little bit, there are two major classes of glutamatergic neuron types in the cortex. One main class is intratelencephalic projecting or cortical projecting neuron types that are distributed across all layers, the IT class. And the other is we call it the non-IT class, which is a collection of different kinds of neuron, glutamatergic neurons types that specifically project into thalamus, corticothalamic projecting, or broadly extratelencephalic projecting, the layer 5 ET neurons. And there are also major glia types, astrocytes and oligodendrocytes that are generated from the same radioglia progenitor cells as the neurons at later stages. And finally, there are the GABAergic neuron types that are grouped into two major classes, the MGE-derived class and the CG-derived class that are generated in subcortical or subpallium ganglionic eminence areas and migrate into the cortex during the development. So our visual cortex taxonomy only captures the cells that have entered the cortex. The GABAergic cell entered the cortex. So from this trajectory tree that we developed purely based on the transcriptomic data, we find that neurogenesis, gliogenesis, and early post-mitotic maturation process give rise to all the cell classes and nearly all the subclasses as the precise timing shown here. And we propose that it's emerged not in a complete inside-out manner as traditionally thought, but really in a staggered parallel manner as shown in the trajectory tree. And we find that furthermore, we find that there's actually extensive transcriptomic heterogeneity in the progenitor population. As the time goes, the radioglia progenitors have dramatic changes in their transcriptomic profiles, expressing already the marker genes, marking the later stage neuronal identities. For example, those genes are markers for the non-IT glutamatergic neurons, and those genes labeling the IT glutamatergic neurons, and those genes down there actually are the glial cell markers. They are already beginning to express in radioglia. And so this is this gradual fate determination process that's already happening very early on and in a what we propose staggered parallel manner. Okay, so then furthermore, we also computationally derived transcriptomic trajectories, not only of the subclasses, but of all the clusters, cell types that we identified in the adult stage. So those trajectory trees are shown here. We were able to retrospectively trace the adult clusters as numbered at the end of the trajectory tree, all the way back to their developmental origins, and also like the branching point of each cluster and things like that. So we have the trajectory tree for all the glutamatergic neurons, the MGE-derived GABAergic, the CGE-derived GABAergic neurons, as well as the astrocyte and oligodendrocyte glia cells. So then here, another observation, conclusion, is that we find more refined neuronal types at cluster level gradually emerge during the extensive postnatal differentiation process from P0 to P28. We see new types emerging at early postnatal stages when the synapses are being formed at eye opening stage, which is right in the middle of around P15, which I'll come back later, and also at the onset of the critical period, which is like at P21. So this are continuous, suggesting continuous cell type diversification during the circuit maturation process. And this is happening widespread across all neuronal types, all the glutamatergic types, especially the IT intratelencephalic projecting, the cortical projecting types, as well as in the parvalbumin and especially somatostatin types, and also in the CGE-derived, especially the VIP neurotypes. Here, I just want to zoom in a little bit more on the parvalbumin and somatostatin types, how they emerge through development. And this constellation, again, is derived from the UMAP showing here. So I wanted to see, so you see many holes in the UMAP, right? It's not a continuous filling of the thing. The holes suggest, so even though the time points are continuous, the holes in the UMAP suggest dramatic gene expression changes. Like cells are jumping across the different transcriptomic space with time. Okay, so here then we want to see how those individual trajectories correspond with the triple modality MET type that we've defined before from our patch-seq data, which are supposed to be well validated because they correspond to traditionally defined GABAergic interneuron types. So here are all the parvalbumin MET types shown here. This is the well-known chandelier cell that's highly distinct from the rest. And then the basket cells in different layers, layer 2, 3, layer 5, layer 6, are corresponding to specific transcriptomic trajectories. Similarly, for somatostatin types, we see these highly complex MET types, Martinelli-like, non-Martinelli-like, different types of subtypes of Martinelli cells targeting different layers. All of them also correspond to specific clusters or group of clusters that have distinct developmental trajectories. And finally, even the intermediate types that we identified before in our patch-seq study that we believe are transitioning type between parvalbumin and somatostatin are found exactly at the junction between the developmental trajectories of parvalbumin neurons and those of the somatostatin neurons, with one particular intermediate type, this layer 6 TH positive type, having an ambiguous origin during development. It was classified as parvalbumin positive, but then if you go back developmentally, its closest cell cluster, it actually mapped to somatostatin. So a true ambiguous origin for this particular type. And then another type is layer 2, 3 basket cell-like somatostatin cell type, actually reflects an interesting convergence. Interesting convergence showing here from the set of clusters originated on a parvalbumin side and another set of clusters originating from the somatostatin side. So we don't understand why this convergence happens, but it's something happens really late during development, probably related to circuit local motif, local connectional motif formation. Okay. So then we focus specifically on eye-opening period because we observe a large number of gene expression changes before and after eye opening, also consistent with previous studies such as those from Larry Zipersky's lab showing new clusters emerging after eye opening. So then, first of all, before we do that, we did a quantitative profiling, profiling of the gene expression changes by summing log-fold changes of all the differentially expressed genes across adjacent ages into this plot, grouped by all the neuronal and non-neuronal classes I told you about. And from this plot, we can see periods during postnatal development where gene expression changes are particularly dramatic. And as you can see here, this includes early postnatal stages corresponding to synapse formation, and this mid postnatal stages corresponding to eye opening. And this late changes, there's not much neuronal changes anymore, but a lot of changes at the non-neuronal level. Okay, now then we focus on this mid-postnatal eye opening period. And this is the UMAP of all the major neuronal and non-neuronal classes I told you about, each UMAP representing a major class. That's colored by the cell types or subclass, and also colored by before and after eye opening period. And as you can see here already, there's dramatic gene expression changes as shown by the UMAP. And now they are quantified here. the number of differentially expressed genes before and after eye opening is huge. There are thousands of genes that changed between those two major periods across all the neuronal subclasses and non-neuronal classes. Interestingly, we find that the neuronal classes, that there are both positive and negative changes, whereas the non-neuronal classes, they are mostly negative changes. Furthermore, among all the genes that change, we observe widespread immediate early gene activation, not just CFOS, but a whole group, 20, 30 core IEGs, immediate early genes. They are plotted here, and they show, many of them show increased expression after eye opening, suggesting that, because immediate early genes are thought to be activity-dependent activation of gene expression programs. So this support the idea that this extensive neuronal activity remodeling after eye opening happening in the visual cortex. In addition to the transcriptomics data, we also generated a smaller set of multi-omic data, also covering the entire transcriptomic, the entire prenatal and postnatal developmental stages, but with sparse sampling. But by combining the RNA-seq and multi-omic data, we begin to be able to identify differentially accessible chromatin peaks that are cell type and age specific, and see how they correspond to the gene expression changes in a cell type and a temporally specific manner. And we see, and this shows the correspondence of peak changes and gene expression changes across all the major neuronal and glial cell classes. So we identified more than 800,000 individual chromatin accessibility peaks in this relatively small data set, actually, just because of the high resolution cell type identification as well as the temporal changes that are captured within this data set. And among those 800,000 peaks, we are able to find all these different modules that have correlated gene expression. Furthermore, by obtaining that information, then we can begin to establish the relationship between gene regulatory elements and downstream targeted genes and derive gene regulatory networks across cell types and throughout development. And we do this by, of course, identifying transcription factor motifs within the peak modules that we identified. And in this plot, it's a complicated plot, but the peak modules are shown in a cell type and age-specific manner here. And then the dominant transcription factor DNA binding motifs within each peak module that also show this corresponding cell type and temporal changes are shown here. And then we plot the specific transcription factor genes that correspond to those motifs and their gene expression patterns. Very often, this motif could be bound by a family of transcription factors. We picked one out of the family that has the corresponding cell type specific and temporal patterns. So by doing this, then we establish the link between specific transcription factors and their binding motifs in the chromatin peaks, as well as the downstream targets that can be identified through the proximity to those peaks, and derive a gene regulatory network, showing how cell type specificity could arise from those putative master transcription factors, and the genes, downstream genes, they regulate either activation or inactivation. Similarly, we can do that and for all the different classes, and this one just shows the GABAergic, all the GABAergic subclasses and the different. We are also able to derive gene regulatory networks across time. So, yeah. Okay, so quickly summarize what I've told you about, and I'll just really quickly fly through the second part of the talk. Actually, it's kind of older study anyway. So from the whole brain taxonomy, We revealed extreme diversity and complexity and distinct features across different brain regions. We find that the transcriptomic types often have regional and more fine-grained spatial specificity. And also the relatedness between transcriptomic types likely reflect the developmental origins and or their evolutionary similarities or homologies. For development, we derived this comprehensive cell type atlas of the mouse visual cortex, reveals continuous cell type diversification throughout the embryonic and postnatal developmental stages. Neurogenesis, gliogenesis give rise to all the major cell classes and subclasses, whereas the increasingly refined types emerge during postnatal maturation and circuit formation process. And we are able to identify cooperative dynamic changes in gene expression and chromatin accessibility in specific cell types and reveal cell type specific and temporally resolved gene regulatory networks. So I think those gene regulatory networks are really important because they show how the gene regulation and cell type specific programs emerge from these coordinated activities of many different genes working together. Okay, so then conversely, we also have another study looking at the aging brain, examining the major hallmarks of aging in a cell type specific manner. We created this data set from 16 broad brain regions, covering about 100 regions in the forebrain, midbrain, and hindbrain, created the cell type taxonomy and mapped every cell identity into our, again, a whole brain reference atlas, covering about 170 subclasses and more than 800 clusters. And using stringent computational approaches, we identified about 2,400 age-associated differentially expressed genes across all these neuronal and non-neuronal types. And many of the differential HD genes are actually now concentrated in non-neuronal cell population with some fewer number of changes in the neuronal population, especially in cortex and hypothalamus. And here, it's just a summary diagram showing the changes, dramatic gene expressions across all the non-neuronal cell types as well as the immature neuronal types. And what we found is the dramatic changes in the cell populations, both neuronal and non-neuronal populations associated with adult neurogenesis. We also find dramatic changes in oligodendrocyte populations, including both the depletion of immature oligodendrocytes in the aged brain, as well as the formation of age-specific clusters that have dramatic gene expression changes indicating deteriorated myelination. We observe widespread increased immune response and inflammation in many different cell types, especially, unsurprisingly, innate immune cells in the brain, including border-associated macrophages, BAMs, and microglia, with all these gold terms of gene expression changes, indicating increased interferon. Interferon signaling, kappa B signaling, inflammatory response, cytokine regulation, all these different things. In addition to the big microglia homeostatic, the giant homeostatic microglia cluster that has all these gene expression changes, we also observed the special age-enriched clusters, 2 small clusters, that have particularly pronounced gene expression changes, again, related to immune response. We also observe widespread antigen presentation gene activation, including both MHC1 and MHC2 molecule genes, not only in microglia and BAM, but also in the ependymal cells I told you about before, tanycytes and other ependymal cells, as well as in vascular cells, endothelial, arachnoid border cells, and finally, even in some special neuronal populations, which I will tell you now. Okay, so then further zooming in, we always started with a big picture and going in. Further zooming in into the tanycytes and the ependymal cells around the third ventricle. So tendicytes has this new beautiful structure with very long processes going from the cell body along the ventricle to into neighboring neuronal structures, properly using those processes to mediate nutrient exchanging, sensing, and regulation. And we see gene expression changes in this area that's confirmed by spatial transcriptomics. And the gene expression changes, again, reflect increased inflammatory response and also decreased neuronal function. Actually, tendicytes have been proposed to also have neurogenic potential. And we also observed increased BAMs, macrophages in this area, especially around the median eminence, suggesting this is hotspot of aging-related changes. What about the neurons in those areas? So this plots all the neuronal changes, with most of the neuronal types with small changes as shown in these gray areas, but a small number of neuronal subclasses showing dramatic gene expression changes with age. And as highlighted here, many of those neuronal subclasses are hypothalamic neuronal subclasses. And where are they? All these hypothalamic neuronal subclasses are concentrated along the third ventricle in the medial hypothalamus, including paraventricular nucleus, dorsal medial nucleus, and the well-known arcuate nucleus that are known to be important for regulating food intake. So here, furthermore, if we go down, not even at the subclass level, looking at individual clusters that are known to express specific neuropeptide and its receptor genes that are important for regulation of food intake and metabolism. We find that high numbers of differentially expressed genes, as well as upregulation of MHC molecules, are showing in specific clusters, highly specific clusters. And what are those clusters? The clusters express in DMH, expressing GLP-1 receptor, leptin receptor, and also clusters within arcuate nucleus expressing AGRP and MPY, clusters within paraventricular nucleus expressing stress hormone CRH, as well as vasopressin and oxytocin that are known to be important for regulating sexual behavior, reproductive behavior, but also now feeding behavior. So just to summarize this study, in the aging study, we found 3 major types of changes: decreased neuronal function, increased immune response, and a hotspot in the middle of the hypothalamus involving both neuronal and non-neuronal populations that are important for energy homeostasis and food intake and metabolism. Okay, so with that, I just want to skip the disease studies to just conclude. By saying, just basically summarizing what I told you about the aging. And really, we're now also aligning, doing the same kind of work in human and non-human primate brains and trying to integrate the taxonomies across age together. And this integrated taxonomy is really emerging. And we're now beginning to able to look at the gene expression changes under a variety of diseased conditions as well. Okay, thank you very much for your attention. I think we're just right. Yeah. Okay. Right on time. I just wanted to acknowledge a large team of scientists and investigators at the Institute leading all these different kinds of work. And I particularly want to highlight the people shown here that are leading the whole mouse brain cell type atlasing work, and a scientist, Kelly Jin, leading the aging study, and Yuan Gao leading the developmental study. Thank you very much for your attention.
Thank you so much.